Okay, let's set the scene. Picture this. It's a really cold Connecticut day, winter 2020. The whole world is basically in lockdown, right? But in New Haven, something pretty remarkable is starting. You've got this renowned Yale professor, Craig Crews, already famous for his work on Kyprolis, a major cancer drug. And he's outside, bundled up with a small team around a fireplace, sketching molecules.
Sketching molecules, it sounds almost, I don't know, quaint, but they were laying the groundwork for a whole new company, Halda Therapeutics.
Exactly. These molecules, called RIPTAC, which we'll talk about more later, they're revolutionary. They don't just, attack cancer. The idea is they literally hold it hostage.
A really different way of thinking about it.
Welcome to BioTales. Today we're exploring the incredible journey of Halda Therapeutics. We'll get into how they're pioneering this totally new way to fight cancer, how they raised over $200 billion.
Right, and secured that huge billion-dollar AI partnership. Quite the story.
And put the world's first RIPTAC therapeutic into human clinical trials. So yeah, it's a story of brilliant science, sheer determination, especially during those tough early days.
And this whole hold and kill idea, it really could change how we treat cancer.
To really understand Halda, you have to start with the founder, Craig Crews. He's got this reputation, a serial entrepreneur who takes science that seems, well, impossible and turns it into actual medicines that save lives.
Definitely. His track record speaks for itself, and it goes way back, late 90s at Yale. He was apparently digging through the Journal of Antibiotics and stumbled across this compound, epoxomycin.
Epoxomycin. Bristol-Myers Squibb had found it, saw it killed tumors, but then just dropped it.
Yeah, they couldn't figure out how it worked. The mechanism was a mystery, but for Crews, that wasn't a starting point.
It was a challenge, a puzzle to solve.
Exactly. Pure scientific curiosity at first. His lab dug in, and they figured it out. Epoxomycin basically gummed up the proteasome.
The proteasome. Oh, okay, for anyone listening, maybe just a quick refresher.
Sure, think of it like the cell's garbage disposal or recycling plant. It breaks down old or damaged proteins. If you jam it up, the garbage, the proteins, they pile up really fast, and the cell dies. And the key insight Crews had was that cancer cells, because they grow and divide so rapidly, they produce tons of proteins. They're constantly churning them out. So they're particularly vulnerable to this proteasome jamming, much more than healthy cells.
Makes sense. And that discovery turned epoxomycin into
Carfilzomib, now known as Kyprolis, FDA approved, a really important drug for multiple myeloma patients.
So from that discovery, he founded Proteolix in 2003 and got Kyprolis into trials in just 18 months. That's incredibly fast.
It really is. Proteolix was eventually acquired by Amgen, a huge success story, all starting from a compound someone else had tossed aside.
But he didn't stop there. His lab then went on to pioneer something called PROTACs, proteolysis targeting chimeras.
PROTACs, these are like molecular scissors, maybe, or maybe more like a tow truck. They grab a target protein you want to get rid of, and they also grab onto the cell's own disposal machinery, the ubiquitin proteasome system, and they basically drag the target protein to the disposal system to be destroyed. Selective degradation.
Okay, so PROTACs destroy, but the early ones had issues.
Yeah, the very first PROTACs were peptide-based, and they weren't, let's say, very drug-like. Didn't work that well in cells. The big shift came around 2009. Crews made the call to move to small molecule PROTACs. He actually said later he thinks that decision saved the whole technology from just fading away.
And that led to Arvinas, founded in 2013.
Correct. Arvinas really validated PROTACs as medicines. Now it's a public company, multiple drugs in clinical trials, a whole new field, really.
So, okay, by 2018, he's founded two major biotechs, Proteolix and Arvinas. He's pioneered PROTACs. Most people would maybe take a break.
You'd think so. But instead, he asked this different question. What if, instead of degrading a problem protein, we just held it in place?
And that question, that's where Halda Therapeutics comes from.
That's the genesis.
And the name Halda, it means to hold in old Nordic languages. It perfectly fits.
It really does. It captures the core idea. Instead of PROTACs destroying proteins, RIPTACs, that's receptor-interacting protein-targeting chimeras, they basically hold them in a molecular wrestling match, stopping them from working.
Let's go back to those early days, early 2020. You painted that picture of the team huddled outside.
Yeah, the world's locking down. They've got 10 people crammed into, 2 offices in this really old lab space. Antiquated, I think, was the word used.
So they literally moved their brainstorming sessions outdoors
Mm-hmm.
to Craig Cruz's patio.
And fleece blanket, sketching molecules on tablets in a Connecticut winter. It sounds unbelievable, but it speaks volumes about their dedication.
Absolutely, just pure conviction in the science. And a key part of that early team was Kat Kaiser-Bricker, who they brought on as chief scientific officer.
Right, a really strategic hire. She did her PhD at Yale, so she actually saw some of the early PROTAC work firsthand. Then she spent a decade at Halda Therapeutics, rose up the ranks to head their early discovery chemistry.
Wasn't that the company Novo Nordisk acquired for over $1 billion?
That's the one, $1.1 billion.
So she left a company that was clearly on a path to huge success to join this tiny 10-person startup working out of cramped offices and, well, a patio.
It was a massive career risk, no question. But her motivation, she said, was being thrilled by the opportunity to invent the next generation of TAC technology with a world leader.
Wow. That really shows the pull of potentially groundbreaking science, doesn't it? Choosing the frontier over the sure thing.
It absolutely does. It's that kind of belief that fuels these breakthroughs.
Okay, so let's talk about the science itself. The RIPTACs, why this approach?
Well, the core problem RIPTACs are trying to solve is resistance. Traditional cancer drugs often target the proteins that drive the cancer, the oncogenic drivers. But cancer cells are smart. They mutate, they find workarounds, they develop resistance. It's a constant battle.
Right.
RIPTACs take a different angle. They don't necessarily target the driver proteins. Instead, they go after proteins that cancer cells just happen to overproduce in large quantities, even if that protein isn't directly causing the cancer itself.
Okay, so they target something the cancer cell makes too much of. How does that work? How does that kill the cell?
Think of the RIPTAC molecule as a kind of molecular matchmaker, or maybe a bridge with 2 sticky ends. One end grabs onto that overexpressed protein in the cancer cell. The other end grabs onto a completely different protein, one that's absolutely essential for any cell, healthy or cancerous, to survive, like a crucial part of the cell's basic machinery. And the RIPTAC forces these 2 proteins together, locks them into what's called a ternary complex.
Okay, so it forces them together, then what?
When they're stuck together like that, the essential protein can't do its job. It's like handcuffing a worker to a bystander. The worker can't work anymore. And if that essential protein can't function, the cell dies.
And the selectivity comes from?
The selectivity comes from the fact that the RIPTAC primarily targets cells that have a lot of that first protein, the overexpressed one. Because there's so much more of that target protein in cancer cells, the RIPTACs tend to accumulate there preferentially. Preclinical data showed something like over 1,000-fold selective accumulation in tumor cells versus normal cells.
1,000-fold. That's huge.
It is. So it's not just targeting cancer. It really is like holding the cancer cell hostage, using its own biology against it.
So why did Cruz pivot? He'd had huge success with PROTACs, the degradation approach. Why shift to hold and kill?
Well, degradation is powerful, but it's not perfect. There are limitations. Some proteins are just naturally resistant to being broken down by the proteasome. Some cell types might not have all the right machinery perfectly lined up. And maybe most importantly, some potential targets are just too vital. If you completely eliminated them everywhere, in healthy cells too, it could be too toxic.
Okay. So degrading isn't always the best, or even a possible option.
Exactly. The hold and kill approach sort of sidesteps those issues. You're not eliminating the protein entirely. You're just stopping it from working correctly by linking it to something else. It potentially offers a wider therapeutic window for certain targets.
And designing these RIPTACs must be incredibly complex. You call them heterobifunctional molecules.
Right. 2 different functional ends, or warheads, connected by a linker. Getting that linker right, its length, its flexibility, its chemical properties is absolutely crucial. It affects how well the molecule binds, how stable it is, whether it can even get into the cells properly. The whole geometry has to be just right.
And there's this other concept, positive cooperativity.
Yes, this is really elegant. It means that when the RIPTAC binds to its first target protein, that binding event actually increases its ability, its affinity, to grab onto the second essential protein. In some experiments, they saw up to a 35-fold enhancement.
Wow. So binding one makes it much better at binding the other.
Exactly. It's like a molecular lock that gets stronger the more you engage it. It creates this self-reinforcing cycle. The more target protein a cancer cell has, the better the RIPTAC works in that specific cell.
Which contributes even more to that selectivity you mentioned.
Precisely. It's a key part of why they can achieve that impressive selectivity for cancer cells.
And the preclinical data published in Cell Chemical Biology in 2024 back this up, showing 50 to 900-fold selectivity.
Yeah, those were pretty striking results, findings that really suggest this could be potentially a paradigm shift in how we think about targeting cancer.
So let's talk about their lead candidate, HLD-0915. This one targets prostate cancer.
Correct. It's the first RIPTAC designed as an actual human medicine. It targets the androgen receptor.
Which is known to be highly abundant, overexpressed in many prostate cancers.
Exactly. So HLD-0915 binds to the androgen receptor, and then it forces that receptor to bind to an essential protein involved in gene transcription.
How does that compare to existing prostate cancer drugs, like enzalutamide?
Well, drugs like enzalutamide work by directly blocking the androgen receptor, stopping it from sending growth signals.
But prostate cancer often becomes resistant to those, right?
It does. The cancer cells can mutate the receptor itself so the drug doesn't bind anymore, or they find other ways, bypass pathways to keep growing.
So how is HLD-0915 different?
HLD-0915 doesn't really care about those mutations or bypass pathways in the same way. It doesn't need to block the receptor's signaling function. It just needs the androgen receptor protein to be present so it can grab onto it and use it to handcuff that essential transcription protein.
So as long as the receptor protein is there, even if it's mutated, the RIPTAC can still work.
That's the idea. And in preclinical models, it showed activity even in models of enzalutamide-resistant prostate cancer. That definitely got investors and the FDA interested.
You can see why. That's a huge potential advantage. And all this groundbreaking work was happening while the team was literally fighting for lab space.
Yeah, those early days sound intense. It was a biotech boom time. Apparently, lab space in New Haven was incredibly scarce. They were in that old, cramped facility that had to be super efficient, really prioritize experiments, make the most of every square inch. And of course, continue the outdoor fireplace brainstorming sessions.
It really highlights how constraints can sometimes drive innovation, like Kat Kaiser-Bricker mentioned.
Absolutely. Focuses the mind. They eventually moved into a proper 9,800 square foot facility, but that early grit, that struggle, it probably defined their culture.
Okay, let's shift to the funding side. How did they get this off the ground financially? Greg Krug's reputation must have helped.
Oh, absolutely. Having successes like Proteolix and Arvinas on your resume means investors listen when you have a new idea. Canaan Partners led the Series A, and importantly, Tim Shannon, a physician investor from Canaan, became the board chairman. That adds a lot of credibility and strategic insight.
Elm Street Ventures also came in early, showing local Connecticut confidence.
Right. They kept things relatively quiet initially, focused on the science rather than generating a lot of hype.
Then came the really big round, the Series B extension in 2024. $126 million.
Yeah, that was a major step up. It happened after they'd shown strong preclinical data for 2 RIPTAC candidates.
And there was a key leadership change around then, too.
Yes. Christian Schade joined as CEO in October 2024. He has over 20 years of biotech leadership experience, things like taking Aprea Therapeutics public, overseeing the Novira acquisition by J&J. Bringing him in really signaled Halda's shift from being primarily a research startup to becoming a clinical stage company focused on drug development.
And that Series B brought in some big names.
6 new premier investors: Deep Track Capital, Fraser Life Sciences, RA Capital Management, Vita Ventures, Boxer Capital, Taiho Ventures. These are top-tier biotech VCs.
Did they say why they invested? What was the rationale?
Joe Cabral from Fraser Life Sciences summed it up nicely. He talked about the urgent need for new ways to tackle drug resistance, and he specifically highlighted RIPTAC's potential for selective killing based on differing protein levels, and crucially, the fact that Halda was developing them as orally bioavailable medicine.
The oral delivery, that's a big deal, isn't it? Especially for potentially larger molecules like RIPTACs.
It's huge. Being able to take a cancer drug as a pill rather than needing IV infusions is a massive advantage for patients. It improves convenience, adherence, quality of life, and from a commercial perspective, it opens up a much broader market. Engineering these complex molecules to work as pills was a significant achievement by the Halda team.
Then came FDA validation. August 2025, HLD-0915 gets Fast Track designation.
Right, for metastatic castration-resistant prostate cancer. Fast track is important. It means the FDA sees the drug as addressing a serious unmet medical need and wants to help speed up its development and review process, get it to patients faster if it proves safe and effective.
And CEO Christian Schade emphasized the highly selective nature and the oral-based aspect as key advantages.
Exactly, underscoring those key differentiators.
But perhaps the biggest validation came with the Vantai partnership, also in August 2025. Potential value over $1 billion.
Yeah, that was a game changer. Not just the potential dollar amount, which is massive, but what it represents is a huge vote of confidence in the RIPTAC platform itself.
And Vantai brings AI into the picture.
Many unknowns. If it works, Halda could essentially own a whole new category of cancer drugs. That's the huge opportunity. But if unexpected safety issues pop up, or if it doesn't show the hoped-for efficacy in patients, that could be a major setback. Not just for this drug, but for the whole platform concept. That's the reality of pioneering work.
Still, the signs are positive. You've got the fast-track designation, that massive Vantai deal, over $200 million from top investors.
And they keep strengthening the team. Dr. Yale joined as Chief Medical Officer in April 2025. He brings decades of oncology clinical development experience. That's critical expertise to navigate the clinical trials effectively.
So it really all comes down to the data now, the clinical trial results.
That's the ultimate arbiter. Did those brainstorming sessions by the fireplace, did all that brilliant science and investment lead to something that truly helps patients? We'll find out.
When you step back, what does Halda Therapeutics really represent in the bigger picture of medicine?
I think Halda embodies a really interesting evolution in thinking. For so long, the main idea in cancer therapy has been destruction. Chemotherapy destroys rapidly dividing cells. Targeted therapies destroy cells with specific mutations. Immunotherapies unleash the immune system to destroy cancer cells.
All focused on killing, destroying.
Right. And those approaches have been incredibly valuable, life-saving, but they all face the challenge of cancer's adaptability, its resistance. The RIPTAC idea offers a different paradigm. It's less about direct destruction and more about, well, control. Holding cancer hostage, as you said earlier, it cleverly exploits something the cancer cell already does, overproducing certain proteins, and turns that into a fatal vulnerability. It's hijacking the hijacker in a way.
And you think this hold and kill idea could go beyond cancer?
Potentially, yes. Many diseases involve problematic protein interactions or accumulations. Think neurodegenerative diseases like Alzheimer's or Parkinson's or even some autoimmune disorders. If you can design molecules that selectively grab onto specific proteins and force them into non-functional or even detrimental interactions, that could be a powerful therapeutic strategy in many different areas.
And combining that flexible RIPTAC platform with AI, like the Vantai partnership.
That creates a really potent discovery engine. You can potentially identify and design new RIPTACs for various targets much more rapidly. It could significantly accelerate the pace of finding new therapeutic solutions.
It's fascinating. Of course, the crucial test, as we keep saying, is whether it works safely and effectively in people.
Always. Clinical success is the only thing that truly matters in the end.
But if this approach does prove out, Craig Cruz and the Halda team will have done it again. Taken something that sounded almost like science fiction and made it a clinical reality, transforming the impossible into the inevitable yet again.
It would be another remarkable chapter in his story and potentially a major leap forward for patients.
So, a final thought for you listening. Consider how this fundamental shift in strategy from primarily destroy to incorporating hold might reshape not just cancer therapy, but how we approach treating disease more broadly in the future. Thank you for joining us on Biotech.
Until next time, keep exploring the science.
Tomorrow's edges in view. A world